Hello and welcome back. The topic for this video, we're going to continue talking about the process of transcription. Now remember, the process of transcription is taking the information that's contained in the DNA in the nucleus and converting it into RNA, all right? Because that RNA has the ability to leave the nucleus. And if it can leave the nucleus, that means it can go out into the cytoplasm, where that information can then be translated into proteins, okay? So with RNA, there are going to be a couple different types of RNA that will be produced within the nucleus. The first type of RNA that we'll talk about is going to be the mRNA. The M stands for messenger, okay? And the mRNA, this is going to be the RNA that specifies a protein. So this is going to be RNA that's going to be important in the process of translation. This is going to be essentially the instructions for making the protein, all right? So that's the messenger RNA. We've also have produced tRNA. That tRNA stands for transfer RNA, okay? And that tRNA, the function of the tRNA is it's going to be what brings the amino acid to the ribosome in our process of translation, okay? So you've got, let me underline these, tRNA, mRNA, and then we've got rRNA, which stands for ribosomal, okay? The rRNA is probably going to be one of the more abundant types of RNA that's produced. And it's abundant because that ribosomal RNA, like the name ribosome kind of implies, is going to be the RNA that's going to make up the ribosome. Okay? So those are going to be the three types of RNA that I want you to be familiar with. There are actually some other types of RNA out there as well. There are some small nuclear RNAs. There's some microRNAs. Don't worry about those. If you know these three for this class, you're good, okay? So know the names, know what their basic function is within the cell, okay? Because those are going to become important next when we talk about translation, okay? But to go back to transcription, because transcription is actually making, the process of making these RNAs, okay? Transcription is going to happen in the nucleus, okay? And we're going to have, oops, a DNA template that's going to serve for our, where our RNA is going to be made from, okay? So that DNA template is going to be some string of nucleotides, okay? Within that string of nucleotides, what you will have at the front, or the 5' end, all right, you are going to have a special set of sequences that's called a promoter, okay? And that promoter is going to be where our RNA polymerase is going to sit on the DNA. Just like when we talked about DNA replication, remember I talked to you about DNA polymerases. DNA polymerases make new strands of DNA. RNA polymerases are going to make a strand of RNA. But in order to make that strand of RNA, that protein, that polymerase, has to know where to start, okay? And that start point for transcription is going to be at the promoter, okay? So it just signals to the protein saying, hey, this is where you want to sit down, this is where you want to start, okay? Then that RNA polymerase will come down, it'll sit on the DNA, it'll physically interact with the DNA, it will read whatever the sequence is, and it will generate an RNA molecule, okay? And that polymerase will keep chugging along that DNA sequence until it reaches another set of sequences. And that other set of sequences is the terminator region, okay? So the terminator is going to be what tells the RNA polymerase, okay, you're done. You can stop making RNA now, all right? So this is essentially what stops transcription. And over here we'll put start transcription. Okay? So if we draw things in here, if we draw an RNA polymerase, the RNA polymerase is going to sit down on the promoter region. It's going to read that DNA sequence, and it'll stop when it gets to the terminator region. Okay? And in terms of that sequence, what that RNA polymerase will do is it's going to put in nucleotides based on the DNA sequence. So just like with your complementary strand of DNA, the RNA strand that's made is going to be complementary to the DNA strand that the RNA polymerase is reading. So in this case, our complementary strand, or our RNA, because we've got A, okay, that RNA polymerase is going to put in a U. When it sees T, it'll put in an A. When it sees G, it'll put in C. When it sees C, it'll put in G. Okay? So this would be the sequence of our RNA molecule. And I have in your PowerPoints that I provide, I've got an example of a sequence that you can practice transcribing. Okay? This is going to be one point where you want to pay attention to how I word things on quizzes and on tests. If I ask you to transcribe, I'm asking you for RNA sequence. Okay? If I'm asking you to translate something, then I'm asking you to make a protein. And if I'm asking you to replicate, then I'm talking about DNA sequence. Okay? So make sure you can keep those straight. All right, in this case, transcribing this would be our RNA sequence. All right, that RNA sequence then would leave the nucleus and it would go based and do something in the cell based on whatever its function is. If it was a messenger RNA, it would go out and it would specify protein. If it's tRNA, it would function out in the cytoplasm, bringing amino acids into the process of translation. If it was ribosomal RNA, it's going to function in creating a ribosome for the cell out in the cytoplasm. Okay? So we've got to start, we've got to stop. So in that process of transcription, really, we've got 3 steps. That first step is going to be initiation. You have to start the process, okay? And you start transcription at the promoter, all right? And this pen's starting to run out, so I'm going to switch colors. Don't let that mess you up, all right? Second step is going to be elongation, okay? That elongation is the process of making the RNA, okay? And essentially it's going to add in nucleotides as it reads the DNA template that it's given, okay? So we've got initiation, the RNA polymerase comes in, finds the promoter, sits down. Elongation, the RNA polymerase reads that sequence on the DNA to make the RNA molecule, all right? And then the last one, probably you'll be able to predict. If not, I'm going to give it to you anyway, all right? Last step's going to be termination, okay? So you have to have an ending for the process as well. Termination is going to end at the terminator sequence, all right? Again, both the promoter sequence and terminator sequences, those tend to be repeating base pairs within the DNA. But I'm not concerned that you know a promoter sequence. I'm not concerned that you know what a terminator sequence is, because there is some variation. There are some different promoter sequences. There are some different terminator sequences. What I want you to know about them is the promoters where transcription is going to start, the terminators where transcription is going to end, okay? So those are going to be the basic steps. For transcription, I think this goes a lot better with some of the animated videos. So within the PowerPoint, I've embedded some animated videos of transcription. I will also provide some links to YouTube videos for you to watch the process of transcription to kind of reinforce that, rather than me continually drawing it up on the board in 2 dimensions, because you have to keep in mind these processes are happening 3-dimensionally within the cell, all right? Takeaway, transcription is going to happen in the nucleus. It's the way of converting DNA to RNA. The promoters where transcription starts, terminators where transcription stops. If you have any questions, please let me know.